Hello, everyone, and welcome to the second of our 6 Cafe Sci talks this summer. My name is Ben Twining, and I am the Vice President for Education and a Senior Research Scientist here at the lab. So before I get started, I want to recognize our supporters. As a nonprofit research institute, the discoveries, solutions, and inspiration that we create are made possible with the help of our generous supporters. And I want to thank each of our donors, board members, and advisory board members for, as our board chair, Bill Burgess likes to say, their time and treasure. I especially want to thank H.M. Payson, who has been a wonderful partner for the lab over the years. H.M. Payson is a Maine-based financial advisor who sponsors our Cafe Sci series each year. So please use the question and answer button at the bottom of your screen to send us questions throughout the talk, and we'll try to answer as many as we can. You'll also notice a raise hand and chat button feature, but those features actually aren't available for this event. So don't get frustrated if you keep trying to push them. So now it is my pleasure to introduce tonight's speaker, Dr. Nicole Poulton. Nicole received her PhD at the Massachusetts Institute of Technology joint program with the Woods Hole Oceanographic Institution, and she did her postdoc here at Bigelow Laboratory and was a research scientist for 12 years and has been the director of the Center for Aquatic Cytometry for 7 years. Nicole is a biologist by training and specializes in phytoplankton ecology. Most recently, in July 2021, Nicole was promoted to senior research scientist. Our senior research scientists here have to raise most of their salaries each year with contracts and grants. And Nicole is currently involved in projects examining the role of phytoplankton in the global carbon cycle, as well as advancing aquatic cytometry methods in a number of ways. And you're going to hear about some of those tonight involving microplastics. I also want to mention that Nicole is an invaluable leader in our educational programs at the laboratory. She has led our high school Bloom program for over 15 years, and she is also a co-leader of our Bloom Educators program, where we bring in high school and middle school teachers and work with them. And she's also been an instructor in our fall Sea Change semester program for undergraduates for many years. She is truly a talented researcher and educator. So with no further ado, Nicole, take it away.
Thank you, Ben. Greatly appreciate the introduction. And as Ben mentioned, I am a phytoplankton ecologist here, and I have been through all that what Ben just described. I've been here for over 20 years at Bigelow Laboratory. And I want to preface this talk with basically saying that I'm not an expert in plastics, but I am very interested in the distribution and the types of what we're going to talk about today, microplastics that are entering all sorts of aquatic environments around the globe, and trying to understand the possible impacts they may have. And I'm going to be talking to you about some tools that I've been using to try to study these microplastics. But before we specifically talk about microplastics, I really want to step back and examine sort of our plastic waste that we have here in the United States in general. And to do so, I explored myself for a while some of the plastic waste produced in the United States. And I started looking also globally and to try to gain a better understanding how we generate and produce a lot of these plastic wastes. So with that in mind, I'm going to start this talk off with a figure. I'm going to talk about how much plastic waste do we actually produce in this country. And really, we have this source from landfills. A lot of this plastic ends up, and some of it gets recycled, as you can see here from this middle picture here. We're familiar with this at our local refuse center. And a lot of it ends up in our oceans and then ends up on our shorelines around the globe. But how much do we produce? So this is a figure. It can be a little bit challenging to understand. So on the y-axis here, we have millions of metric tons. And we have from 1960, this is looking over time, to 2018, and which is the most recent data from the United States Environmental Protection Agency, looking, and this is a report from December 2020, and it looks at our total municipal solid waste generation in the United States. And in specifically 2018, excuse me, I'm going back, in specifically 2018, the overall municipal solid waste that we generated was 292 million tons. Now, that's a large number, and it's really hard to sort of grasp how much that is. So I put it in terms of the size of an aircraft carrier. So an average aircraft carrier weighs about 100,000 tons. So that is equivalent to about the weight of trash, or waste in this case, of 2,920 aircraft carriers, to try to put that in terms of a context of how much waste we produce. Now within that, in 2018 specifically, and that's right here, this is the plastics that we generated, the proportion of trash that was actually plastics. And that was about 36 million tons. And a ton in this case is a British units or English units. So these are 2,000 pounds per ton in this case. So the waste that we generate has many different aspects to it. It includes food waste, yards waste. We have metals, glass, and a lot of paper that ends up in the trash. And this is basically to give you a sense of how much we actually produce. Now, of that plastic, the 36 million tons that's generated, a very little amount in this case, in 2018, if you think of the total amount, 36 million tons in 2018, only 9% down here basically is recycled, or what we define or we think is being recycled. The remainder of this plastic is either burned or is buried in the landfill, as per the US EPA. And however, it does not include a lot of things. So if you think of clothing or textiles and all of the potential illegal dumping that's happening, including litter, the trash that we see on the streets or that will end up in the ocean eventually, that is not included in this estimate. And about half of the recycled plastic that we put into that blue bin that we always see is actually shipped overseas to be treated or recycled in some way. So as a comparison, I just wanted to think of both the US paper generation and recycling and we have a better system for recycling paper in this country. So in 2018, about 67 million tons of paper or waste were generated. And about that, almost 70%, 2/3 of it was actually recycled. And only about 6% was burned and about 26 ended up in landfills. So the types of plastics that are in our solid municipal waste, and this is specifically looking at the most recent data that we have, and this is from 2018. And it's a variety of different things, but predominantly, if you look here, it is predominantly containers, packaging, bags, sacks, and specifically bottles that we're most familiar with, but disposable bottles that are not, only have a very short lifetime, and they end up either being hopefully recycled. And in this case, many of these bottles are predominantly recycled. And this is your classic milk jugs, as well as your drink bottles, your freshwater bottles. And included in that are also, of this 36 million metric tons that are produced, we also have non-durable products and durable products. And the non-durable, just to put it in context, are those things like disposable cups, silverware that we use very, very short term. In this case, less than 3 years. And durable products are those things that are quite heavy. So that's why it comprises the majority of the weight of our waste here. And this is refrigerators, microwaves, things like that, that would end up that have a lot of plastic associated with them, but they last a lot longer. They're about 3 years longer, 3+ years. So I'm also going to talk about a science study that just recently came out at the end of last year. And this was a study by Lavender Law et al. And it was basically described the solid municipal waste that the US had generated in 2016, specifically plastic waste in 2016. And at the time the study came out, this was really the most up-to-date information that they had. And they looked at all the countries, or many coastal countries around the globe, and they estimated that the United States is the #1 generator of plastic waste in the globe, on the globe. And this is based from 2016 data that's from the World Bank and the data, similar data that I just showed you in terms of the US EPA estimates. And this is the managed waste that they were calculating. But what they took into account in this study was waste estimates that had not been previously taken into consideration, specifically illegal dumping, mismanaged waste, and the recycling that takes place where we think it's moving overseas. That's half of our recycling ends up overseas. And then littering in general. So what it showed was that, as I said, the US is the #1 waste generator. And this was over 42, they believe 42 metric tons in this case. And of that, the US waste, plastic waste, many of it ends up in the coastal environment, and that can range from 0.5 to 1.5 million metric tons. Again, putting that in context to aircraft carriers, for example, we can have 42 metric tons is about 420 aircraft carriers of just plastic waste. And of that, a single about 14 aircraft carriers in terms of what ends up going into the coastal environment. So again, this is a lot of mismanaged waste that is actually taking place here. And some of that mismanaged waste is going overseas to countries that also have problems managing their waste. So trying to understand now, moving from the United States to a more global perspective of plastic production in general, I wanted to give you a sense here that global plastic production has been increasing over time. And there's been 3 phases of this global plastic production. So we're starting here in this graph, we're looking at very early in our century here in terms of like the 1915 going all the way to 2015. And we're seeing phase 1 here is signified by slow development and invention of plastics that are commonly used today. And so this is the innovation phase of plastics. And then in around World War II, at the end of World War II is when they really sort of ramped up production. And this is what we call the global and rapid global expansion or growth phase of plastics. But now we're in this final phase here. The production of plastic is mirrored by economic growth, which is also indicative of also population. And in 2015, the types of plastics that are being produced for different economies here, we think of like packaging, consumer items, also construction, automotive, and electrical and agricultural products. So in 2015 alone, we had equivalent to about the weight in this case of 3,250 aircraft carriers of just plastic being produced globally. That's a lot of plastic. So how much plastic actually enters the oceans of this plastic? So there's a study in 2015 that looked at this. This is a science by Jambeck et al. And they just looked specifically at at 192 different countries and looked at the global waste production for that one particular year. And this was in 2010. So I'm going to walk you through this figure a little bit. So again, 270 metric tons were produced of plastic globally in 2010. And this is 275 metric tons were actually produced as waste. And so what the overall goal here is like how much of this plastic that we're producing and then creating waste is ending up in our global oceans. So how they calculated this is that they looked at the distance and the amount of waste being generated within 50 kilometers of the coastlines of these 192 different countries. And in that, they were able to find or determine, that about 100 metric tons is produced within that 50, going backwards here. It's produced within that 50 kilometer span of distance from shore. And of that, about 32 million metric tons is mismanaged. And so that is the key process right there, because mismanaged waste can end up going into the oceans. And they estimate that around globally, across the entire globe in 2010, about 8 million metric tons was going into the ocean. And the study goes on to talk about how this potentially could exacerbate over time and increase rapidly, not so much exponential, but as population growth increases, we're going to see that number, 8 million metric tons increase over time. So what types of plastics end up in the aquatic environment? We have a variety of different types, and I talked a little bit earlier about drink bottles and milk jugs. And so they have many different chemical constituents here. We have polypropylene, polyethylene, but specifically what you recognize them as is milk jugs. And we also have a lot of plumbing and construction plastics that make up a majority of some of these products. And so we've moved from a lot of metal to a lot of PVC, or these plumbing plastics. We also have, we also have packing and insulation. That's actually think of all the plastic and styrofoam containers that we use for most of our food. And in many cases, this is also insulation. So this is another big component of all of the plastics that we produce. And this comprises about 80% of most areas around the globe of the plastics that we produce. So I'm working towards microplastics, which is what the focus of my talk is on. And as things start to enter the ocean, they can come in at various sizes. And so a lot of size classes of plastics. And so we're talking about the smallest component of today's talk. But these larger macro-sized or large plastics are these first two groups here that I'm going to be talking about. So these are the things that you can see with the naked eye. So we've classified them here. These are the ones that are very large, that float on the surface and potentially could end up on a beach. And then we have smaller pieces of plastic that are broken down and actually potentially consumed by, in this case, you can see here, this is, it's been impacted by a bird. But the plastics that I'm focusing on in today's lecture are primarily the really small plastics that, in this case, in fact, can impact much smaller parts of our aquatic food web. In particular here, we can start with these ones going with the smallest organisms, which first very similar, like which I study, are plankton and bacteria. And those are consumed by the small grazers, such as larval organisms. And you can see a picture here of a larval fish. And inside, they actually have microplastics, in this case, actually microbeads. This was done during an experiment, most likely. And they can actually see that these larvae can actually feed on these microplastics. And this can move up the food chain and impact turtles, seabirds, mammals, and eventually top predators like ourselves. But specifically the plankton is what I'm most interested in. And here's an example of some feeding experiments where people have looked at the impacts on, this is a zooplankton called the copepod, where they're actually consuming, and if you were to look very closely here, those are microbeads that you can actually see inside the gut of the zooplankton. So some of the sources of these smaller microplastics and possible impacts of these, it's like, so there's two sources of microplastics. They're actually produced at the size that we're talking about. And these are you're familiar with the ones that are now currently banned in things like toothpaste, the scrubs, the abrasives that you use in your day-to-day products. And these are your facial scrubs, sometimes your shaving creams, but in a lot of cosmetics as well. So they're actually made small, these plastics. And the secondary sources are things weathering. There's lots of weathering that takes place of things from shore, like we have laundry, your clothing weathers, you have fabrics that are your fibers that are coming off your fabrics, laundry, as I said, weathering of plastics, and from wastewater treatment plants, which is what WWP stands for, TP. And this is all entering the freshwater and marine water systems. And what sort of impacts they can have? A variety of different impacts. This can range from, so these small little pieces of plastics can serve as biofilms for lots of microbial life. And that can be either a good or a bad thing, because we're hoping that we can begin to find things microbial that can maybe biodegrade these microplastics. We can also look at reservoirs and pathogens that are actually being transmitted via microplastics, which was not a good thing in that case. We've already seen that they can be ingested by smaller plankton, which would then be ingested by larger fish, for example, or filter fed, like a lot of fibers. We can see, and there's been some studies even done here at Bigelow about fibers being absorbed into, or not absorbed, actually fed on and taken into the gut of some of the shellfish. And a lot of these microplastics can also accumulate pollutants on the outside or the exterior of these microplastics. So these are ways for things to move up through thinking of bioaccumulation through the food chain. But the secondary sources of microplastics are primarily coming from the larger plastics being degraded either physically. So we have the ocean wave actions. We also have oxidation. A lot of plastics are very sensitive to UV light. So what we find is that over time, and where these microplastics form is just that they break down fairly quickly, actually, as we are learning. And so they form these fragments, and these fragments then become reservoirs for bacteria, where hopefully we begin to find, as I said, biodegradation, ways of basically bacteria that will consume the plastics as a carbon source and actually degrade these plastics. And that's what we're hoping to find. So with that, I'm going to pause here for a series of questions from anyone from the audience. And then what I'm going to focus on in the second half is a case study where I am and how we study microplastics in the aquatic environment. So at this point, I'd be willing to take any questions from the audience, and you can use the Q&A function.
All right. Can you, there I am. Hi, Nicole.
There. Hi, Ben.
So great. Yes, we have a couple questions that have come in that I'll read to you. The first is about, you talked about mismanaged plastic waste. How is that defined? What makes plastic waste mismanaged?
So when we think of what the US EPA actually reports on is managed waste that actually ends up, and they've measured it, and this is from municipalities all across the country, for example. They bring in this waste and they can quantify it, but what the mismanaged waste is also from illegal dumping and litter. And so it's the waste that is not accounted for in a lot of what the US EPA is generating in terms of their reports, and which is a very common thing because we have seen waste all the time around the globe. So any other questions?
Yeah, sure. What do we know about the relative abundance of the various size classes of these plastic wastes, right? You talked about the 4 classes. Do we know if one of those is like more abundant than the other?
Well, abundance is there's weight and then there's abundance. So in terms of weight, there's going to be much more in the larger size class because they haven't broken down yet. So when they're in the ocean and they're sort of floating around and they actually contribute some more of the weight because they're still intact. But as they break down, their abundance starts to go up. However, what you're going to find here in the rest of my talk is that it's very challenging for us to enumerate and understand the distribution and the amount of these smaller microplastics. And now what we call even another size class, which is even smaller than bacteria, which are these nano-sized particles. And those are the ones that are even harder to analyze. So we're still in that discovery stage and trying to understand the actual abundance of these particles.
Great, thank you. Okay, let's see. How much of the macroplastic waste is fishing gear? Do you have a sense of that?
I would have to probably look that actual number up. However, the fishing gear is one component of it, but I would say that what's making up the majority of the plastics in the ocean are coming from land. So this would be your coastal waste in terms of bags, not those non-durable goods, the plastic cups, the plastic forks and knives, things like that, that are coming directly from landfills that are actually quite close to the ocean.
Okay. Here's another one. Once macro and microplastics are in the water, freshwater or saltwater, is there any way to collect them?
It's a good question. And in the research that I'm going to be talking about in the second half of my talk is specifically to collect plastics for research, but it would be great to find ways to actually accumulate on the surface, or try to collect this plastic. And now, from what I have been reading, one of the most important ways of actually managing waste is actually beach cleanups, because a lot of that macro or large plastic ends up back on a beach. And that's actually the best way of collecting it, because out in the open ocean, it will photo degrade over time and then form rather rapidly into these smaller microplastics. So it's better to catch them early. So this has been something that's been recently found out by some folks, some colleagues in Europe, actually. So it's one way of actually managing that. But there are companies I know that are looking into ways of gathering the surface plastic, but we have the 2/3 of the planet is covered by oceans. So it's going to be a huge challenge no matter what we do.
Big surface. All right, let me do a couple more here. So besides the problems associated with food chains and pollutants, do we know much about how waste plastics in the ocean might limit light availability for phytoplankton? There's a good question for the phytoplankton ecologist.
So as I'm about ready to talk about, the concentration right now, I mean, we don't want them to get too much more abundant, but at the same time, right now, the concentration is so low that we have to concentrate or bring these microplastics together even to study them. So at this time, they are not problematic for phytoplankton growth as far as I know. However, in the laboratory, we've done experiments, and we have actually colleagues here at Bigelow, Patty Matrai, who have actually done this to look at phytoplankton, but more from a toxin perspective. But if you get the particle density high enough, it will limit the light for the phytoplankton.
Yeah, right. Okay, there's a couple of questions here on the same theme, so I'm going to group them together. And essentially, you mentioned that there might be microorganisms or bacteria that could potentially break down plastics in the ocean, and I'm wondering, there's a lot of interest in that, and if you can say anything more about whether those have actually been identified and whether we know if they're making a difference.
There are a few review papers that have been coming out in the last couple years, and there are bacteria that they have now identified that actually can use certain plastics as a carbon source. So it's a slow process, but yet at the same time, they're finding pitting. So they've done it in the laboratory and have started to isolate these organisms. So you could think that down the road, there may be some advantages to knowing that there are plastics that can be biodegraded, and I'm not sure if this is for all plastics, but I know there's quite a few of them that actually have been identified, certain bacterial strains that can degrade them.
Cool. We love the microbes here at Bigelow, so I'm glad to hear they're doing their job. So great, we'll let you keep going. There's a bunch of other questions, but I'll hold them until the end. And
okay.
Audience, please keep sending them in. And yeah, Nicole, back to the show.
Okay, now picking up on that thread, how do we collect microplastics? So I've been talking to you about, and we just discussed in the Q&A, that they are very abundant. So, and as I think I just mentioned, the challenge is that microplastics are really dilute right now in aquatic environments. So it's real, I mean, for phytoplankton, I can have thousands and thousands in a cubic centimeter of water. Whereas here we can have, we're talking about 1 or 2 in a cubic centimeter of water. So in this case, we have to concentrate this to these microplastics to be able to study them and to understand their impacts. So we have multiple ways of concentrating water, and we're very big professionals at concentrating water because I do a lot of phytoplankton research to be able to concentrate and analyze different chemical constituents within phytoplankton. So to do that, we do for surface, and this is for some of the larger microplastics, we'll use things that, because microplastics can float on the surface of the water. And this is what we call a certain area right here at the surface. And this manta trawl, it's just because it mimics a manta, because it looks like it has wings, and that's what keeps it at the surface because it's floating there. The manta trawl basically will take a subsample of just the surface water, and it will concentrate what's at the surface. So they can do a long tow. They'll know how far they traveled and how much water passed through, or how much surface area passed through the net, and they can concentrate the number of plastics. We can also use pumps. So this is a pump made by McLean Labs. Actually, this is down near the Woods Hole Oceanographic Institute. And these are pumps that are traditionally used to look at microbiology in the water. But in this case, the focus is microplastics. And these can be done at different depths. So it's a submersible pump, and it can filter the water. So how do we separate and identify these different microplastics? So again, we're talking about things that are very, very small. And so when I say micron or micrometer, the way of putting it into perspective, think of the width of your hair. So the width of your hair is approximately, depending on if you have thin hair or thick hair, about 100 to 200 microns in size, the width of it. So, 1 micron is 1/200th or 1/100th of that width. So we're talking about very small particles. So to do this, we have to concentrate, which I just described using, in this case, this jar here is from a manta trawl. This has been collected off the surface, and they can dry it and then they can weigh it. And that's one way of looking at mass. But we have to first remove a lot of that living material. We don't want the phytoplankton, the wood, the leaf litter that goes into this trap as well. And we can oxidize that away. We can get rid of it chemically. And so we can weigh it. And some of it, we use density separation. We can actually remove that surface layer, and some of that plastic does float, not all of it though. And then we spend a lot of time under the microscope. Not myself, but a lot of colleagues at NOAA do this as part of their marine debris studies, and they count and characterize it, what type it is, meaning like shape, color, things like that. And then the plastic can actually be identified chemically and analyzed. This is great for large microplastics, things that you can see with the naked eye, or like the small microbeads that you see in abrasives. But it's not possible really for the really small things that I was talking to you, that's down in that 1 micron levels size range, where you're thinking again, the width is smaller than the width of your hair. So how do we count and characterize these small microplastics? And so I use 2 tools. We can use microscopy and something called flow cytometry. And this is where I came in to help people that are using or studying microplastics. So here at Bigelow, we have a Center for Aquatic Flow Cytometry. And I'm going to talk to you a little bit about what flow cytometry is, so that you can see how I'm using this to characterize microplastics. So flow cytometry really is just the study or detection of suspended particles in fluids. And it was developed quite some time ago, actually in the 1970s. Now the instrumentation has gotten a lot smaller over time because the computers have gotten a lot smaller. And this was developed in the biomedical field. And if you actually go to the doctor and you have a blood sample drawn, you actually have it most likely analyzed by a flow cytometer. And that's looking at different red and white blood cells, for example. But in my applications, I can use the same tools that are used in my biomedical applications, but for studying things like phytoplankton, bacteria in the ocean, and we can tag them in some way and make them light up. And in this case, I can also use it for these small microplastics and actually even separate them from the sample at hand. So what I've been developing here with some help of some Colby College students over the last few years, we've been using a method that's used in microscopy, but it's called tagging the microplastics. I have to make them light up in a sense. And we use a stain called Nile red. And this is how it looks under the microscope. So they've actually been using this method to look at these really small microscopic microplastics. And to do that, on the left-hand side here, this is just what a standard microscopic image would look like of these different types of microplastics. These are abbreviations of the type of plastics. So polypropylene, polyethylene, polystyrene. So if you think about this, this is sort of like what your water bottles are made out of, and this is what your styrofoam is made out of down here. And once you've tagged that plastic, it can light up under a certain wavelength of light. And so what you can see here is that glow that we're looking at. And so this is a tool now that I'm developing. Rather than it being a microscopic method, we're using flow cytometry. So this is just this is the stain that I use to tag these microplastics. And this is what these from a field sample look like under the microscope. And so the red Nile stain adsorbs, attaches like some chemical pollutants do to the surface of the microplastics, and it causes them to glow under laser light. So I take this concentrated particle mixture and I put it into a flow cytometer. And you can see here, this is the concentrated mixture that I've made, and some of the particles are plastics and some of them are not. So these green ones that are basically stained with Nile red are being individually analyzed as they go through the instrument. And we can do that. This is a Nile red stain that's attached to the surface of this microplastic particle. And so individually they're going by, but what's the advantage of flow cytometry is that this is happening at a very fast rate. These particles are going through the instrument at hundreds of thousands per second. So it's something that's happening at a very fast rate. And then we can isolate those particles. And so these are the ones of interest here, and these are the ones that are tagged with the Nile red stain. So how do I use this in my laboratory? So I have these concentrated particles that are from the field, and then I can detect the size of the particle and that glow of the particle. And I can plot it here, and where it has a lot of fluorescence, I can actually isolate them and count them. So this is a way of enumerating these really small microplastics that are in these field samples. And here's an actual picture of real data to show you. And each dot represents a particle that's gone through the instrument. And I can actually, this is a polystyrene bead, and I've been able to count it and separate it from the sample. And then I can, at this point, take the sample to be chemically characterized. So with that in mind, I've done this here at the Center for Aquatic Cytometry, and we've been using and testing it with plastics of known size and shape. So in this case, it's a bead that we know of. And then in this sample here, this is from a Colby College student that was working on this with me. And these are samples of field samples that have tagged microplastics in them. So where we're applying this is in a case study, and this is taking place in Lake Superior. And this is one near and dear to my heart, mostly because I grew up in Michigan at one point, and I have some colleagues at the University of Minnesota in Duluth, and they came to me with actually this idea of testing this. And I said, I'll give it a shot. We'll try it out. It works really well for phytoplankton flow cytometry. Let's see if we can make it happen with microplastics. So the largest of the Great Lakes is Lake Superior. And this is also the one with the largest surface area of any freshwater lake in the world. So it's, I used to camp on there when I was a kid, and you can't see across it, and it looks like the ocean in many cases. And it has about 10% of all the fresh water on the planet. So my collaborators at the Large Lake Observatory at Duluth, Minnesota. And the goal of this project is really to characterize these very small microplastics in Lake Superior. And we're doing this in a specific area of Lake Superior. And this is Minnesota Duluth area in the western arm of Lake Superior. Just to give you a sense, the length of Lake Superior going from one end to the other is about 350 miles. So we're talking, it's a very large lake. So microplastics, so to study these microplastics in Lake Superior, my colleagues have been using the same tools that I just described, both the submersible pump and the manta trawl, to look at different size classes, as I've discussed with you, to look at surface microplastics as well as microplastics at different depths. And we've divided them based on their size. So we've divided them up, and they do this from a boat. This is the RV Blue Heron that they go out on, and they're doing this at multiple stations. And here we are, we have multiple filters that we're using to collect different sizes of these microplastics. And so we can concentrate them down off each of these filters, and then they are sent to me for this tagging method so that I can analyze them. In Lake Superior, we have multiple field sample locations. And so right now they've been sampling these yellow dots that you can see here. So this is a deeper station way offshore. Duluth is located right here at the end of the western arm of Lake Superior. And so at these different stations, they're also doing multiple depths. And there's a survey that was done in May of this year, and is currently taking place this month. And I hope to be doing over the course of later this year and into the spring, some of this microplastic analysis, where I will be separating the microplastics from the samples and shipping them to her for chemical analysis. So what are the overall goals of this project? Really, the idea is to really understand, because there's so limited knowledge. I mean, there's so much limited knowledge in the ocean, but this is really in a lake where there's a lot of runoff from land, especially from wastewater treatment plants. And so there's a lot of potential plastics entering the system. And it's also a big source of drinking water. And to understand the prevalence of these, we need to really understand the prevalence of these different plastics in Lake Superior, because really there's not been many surveys done. And my colleagues are really one of the only people that have been looking at this. And they've been recently funded by a Minnesota Sea Grant to do this work. And it's really to produce methods to be able to quantify and characterize these smaller constituents of plastics in aquatic environments. So it's a big issue for the state, both Michigan, Wisconsin, and Minnesota, where Lake Superior borders, and looking at drinking water contamination, as well as food web impacts in the local environment. You know, it's a big fishing destination as well. And so we're really committed to try to understand the source, so we can start to look at effluent coming from wastewater treatment plants, the effects, and potentially the fates of some of these microplastics in these environments. So I want to just sort of end on a high note as much as we can, given the fact that I've talked so much about plastics and how much we've produced these plastics, as well as potentially the issues that we have with these microplastics and trying to understand their concentration, their abundance, their distribution, where are they and what are they? So I'd like to say that there's a variety of different things that we can do. And as consumers of plastics, it's really reducing. I mean, this is sort of a no-brainer to many of us to basically reduce, reuse, and dispose of responsibly. It's really understanding where is your when you go to recycle something, where is it actually going to be recycled? So it's just trying to understand that process and being more aware of where your recycling is taking place. And that's from the consumer aspect. So where we can think of other things that we can do, and you can think of the state or the government legislation area, is really the goal overall is for waste management and to have less or zero waste in terms of plastic. And in terms of governance, you can think of like limitations. I mean, I think of the state of Maine, for example, and we now no longer in the state of Maine have plastic bags as an option when you go to the grocery store. So that is one incentive or one regulation and legislative process that's actually trying to reduce our impact. So with that, I'm going to conclude and thank a lot of the people that make this possible. So here at Bigelow, we have a small pool of money that we can access called the Spencer Entrepreneurial Fund. And this has really been helping me develop this microplastic detection method using flow cytometry. And the Minnesota Sea Grant is really what funded my colleagues, both Elizabeth Miner and Melissa Mora-Jones at the University of Minnesota. And they were able to really get this started, and I'm helping them to analyze the smaller end of their microplastic size range. And I have to thank many members of my own lab, Lori Lebelzik, who's helped me throughout most of this and runs a lot of my flow cytometers in the lab, and 2 students, Charlie Cobb from Colby semester 2020. Actually, no, she graduated in 2020, and Taylor Kruger, who just graduated this past year in 2021. And they both had independent research projects that were focused on microplastics. So they helped immensely. So with that, I'm willing to take any final questions, and I'll pass it over to Ben.
All right, great. Thanks, Nicole.
Mhm.
And exciting. Always happy to see students involved in research. That's a big theme here, and one that
Yep.
so awesome. Okay, we've got a couple questions here. So there's some really interesting ones. So here's a good one. Is the increasing severity of coastal storms affecting the amount of litter in the ocean? Any idea if people have looked at that?
It's a very good question. And I would say that I wouldn't be the best person to answer that question, but my guess would be yes, because it's really, I mean, not only is there just mismanaged waste at the coast, but you think of landfills that are near the coastline, and if they aren't mismanaged, if they're not managed properly, then more waste will be going in. I mean, you think of just in general, especially for the microplastic work, you think of river flow into so if there's a rain event or storm event on land or near the coast, that's going to have a lot more runoff. And the runoff is a source of microplastics from drainage from basically roadways, as well as not so much wastewater treatment plants, because that's a regulated outflow, but basically river systems would be a source that would go in. So my educated guess would be yes, but I would have to ask a specialist.
Yes, but good. Here's another one. We've heard about the sort of plastic islands. I think meaning, these regions where plastic sort of accumulates in the surface of the oceans. You know what portion of plastics in the ocean are in such of these regions, as opposed to plastic that might just be widely distributed,
Mm-hmm. So in some recent studies, I would say in these areas in the center parts of the ocean, that is where a lot of microplastics accumulates. And that's mostly due to circulation patterns of the global, and you think of the Gulf Stream, Kurosha Current in the Pacific, those areas, and they form like, in a sense, that currents go around them, and they form these areas called gyres, where these sort of more static water, and that's an accumulation point for these sorts of things. But what we're also learning is that these larger plastics, and through studies of ocean current modeling, is that a lot of the larger plastics that end up the ocean do end up on the coastline. So if we can catch or reduce or capture plastic that's large before it becomes microplastics, you can probably do that closer to shore than we can the microplastics, which will end up mostly in these gyre or center of the ocean regions where there's less movement of water.
Okay, great. So here's another interesting one. We might need to call in Kath from last week for this one. So recent papers have stated that microplastic concentrations can be estimated from satellites. Smoothness of surface is affected and measurable. What do you think? Ever heard of that?
So yes, I have been, it's a potential, yes. That from what I've heard, and that's not my area of expertise, but Kath Mitchell, who spoke last week, who works with satellite data and is very interested in actually pursuing this. So I'm hoping that this might be a way of actually at least looking at the distribution. Now we're talking at the very surface layer of the water. So this wouldn't be looking at anything at depth, which there are components of the plastic that do sink or believed to sink, and that would actually be a fate for some of these microplastics to go at depth. So it's still questionable, but yes, I do believe that there is some promise into the study of some of these microplastics from satellite data.
Cool. All right, I'm going to group two questions together here again. So one person asks, has anyone studied microplastics in airborne dust as a source to the ocean? And sort of related to that, I think, is someone asks about burning plastic and whether that produces microplastics. I mean, since so much, I think, of our plastic waste is eventually incinerated, do you know if that could turn into microplastics? And if so, does that go into the air and get to the oceans?
So it is actually a newer part of the component to study microplastics. We've been so recently been focused on microplastics in the aquatic or coastal environments or open ocean environments, but there actually is now research where, I mean, microplastics they're so there's light, so when they get small enough, they actually can go into the atmosphere, especially fibers, especially come from textiles and things like that. So I have actually read in some papers that is now being a contributing factor. So they actually do go into the atmosphere. Now, as for burning, it really depends on how efficient burning process is, I would say. So I'm not 100% on that as a factor for actually inputting plastics into the atmosphere, but we should check it out.
Okay, great. All right, here's one that is near and dear to my heart because I came across this question in the lab today, which is, could you envision developing home testing kits so that we could collect and measure microplastics in our waters as a form of citizen science? And I will add, today in one of my cultures, we saw some strange floating white bits that I think were plastics, and we spent a lot of time looking at them under the microscope, wishing we could clearly identify them as plastics. So what do you think about this as a possibility, Nicole?
It's a good question. Plastics in general, I mean, even though I talked about the impacts of them and moving up to the food chain, and if anything, they could be, bringing adsorbing toxins to their surfaces, but they're relatively inert. So the way I chemically characterize them, in some cases, you actually have to burn them to actually chemically characterize them. So I'm not sure if there would be a good or effective method like a home test that would be, but then I'm not a chemist, so I can't justify that exactly, but that would be a guess on my part. However, I think the second part of your question was, we talked about the home kit. What was the other part you said?
Well, it was just my own observation that I wanted such a kit.
Oh, right. Yes. Well, we would all love that. But what's really interesting is that there are a lot of, actually what I was going to talk about was, working with these microplastics, you have to be actually super careful because just wearing a fleece that's basically has huge fiber components, you can actually see these plastics in your own samples. So if you actually have to use glass for everything and make sure that you're wearing cotton usually, but even that produces fibers. So it is rather challenging to actually identify these things, but the removal of any sort of organic material like cotton could be removed in some of the processing of these samples.
Interesting. Do you see any shifts in plankton ecology in areas with more microplastics?
Right now, I mean, I'm at sort of at the very beginning of this type of research in terms of the impacts, but when you take microplastics and actually expose them to much higher concentrations of plastics in the laboratory, there are some impacts. And this was done by some former REU students here at the laboratory. So, and we've also seen that they can have positive and negative impacts. So whatever these microplastics might be bringing along with them could actually be benefiting growth, or it could be causing a negative impact on growth. So TBD.
TBD. Okay. All right, we've got two more here, and then we'll let you go. So one person asked about plastic balloons, and I know that's something as a parent of small kids. Do you have any sense of, the data you showed in the beginning, if those are an important source? Is that something we should be worrying about?
It's one of the many sources of plastics, and because they can, let a lot of helium balloons can end up in the sky. They can definitely end up in the aquatic environment very rapidly. So that's one aspect of that litter that I was talking about in my talk in this paper, that's identifying other sources of this mismanaged waste. And that's one component of the litter that goes into the ocean. So it's hard to say. I mean, I think there's a lot more drinking bottles and, plastic disposable cups and things like that that are actually potentially more of an impact, because we have plenty more of those in, given our society and food and the food industry and things like that. But I'm sure balloons is a huge impact.
So great. So thank you. We're going to end on one other that is sort of on the applied side and sort of the what can we do?
Yes.
And that's asking about, the bottle deposit. We have this 5-cent bottle deposit in Maine. And you mentioned the ban on single-use plastic bags that Maine has passed. How about, have you heard discussion about trying to increase the bottle deposit? Do you think that would have any positive impact on, sort of the litter of single-use drinking water bottles everywhere?
I think it would. Yeah. I mean, I think more people would return bottles that way. I mean, it would be also a way of nonprofits making more money, since they have all these collection sites all over. But yet at the same time, I also feel as though it would be an impact, and those are other ways. And those types of bottles, especially milk jugs unfortunately don't have a deposit on them, which I sort of feel as though maybe if we could add to the types of plastics that could actually carry a returning fee. I mean, because that would be, like all the milk jugs would then be guaranteed to be returned. So things like that. And because those are plastics, especially the drinking bottles, that are more easily recycled here in the United States. About 30% of the drinking bottles and milk jugs are actually recycled in the United States. So that would encourage that to take place.
Great. All right. Well, thank you so much, Nicole. I know I have learned a lot, and I really appreciate you working on this really important topic. So thank you.
Thank you.
For the rest of us, please join us next week to hear from Dr. Alex Michaud, who is a research scientist at Bigelow. Alex is actually going to join us remotely from the Tulik Field Station in Alaska, which will be an interesting feat of technology. And he's going to talk to us about his work to understand the important role that microbes play in the ice environment. He's going to take us on a journey across from pole to pole to understand what's going on in these rapidly changing environments. And with that, I want to thank all of you for joining us tonight and for caring enough to learn about the ocean and what Bigelow is trying to do to protect it. So I hope to see you all next week, and have a good evening.
Thank you.
Thank you, Nicole.